Assalamualaikum and hello to Dr. Nur Hidayah and my fellow classmates. So in this video, what we want to share with you is about the newest technology of this era, which is called microarray technology. Now let's get started.
Assalamualaikum and hi everyone, I'm Nurfatihah and I'll be presenting the history and introduction. So we will first look into the history of microarray. The early history began with the discovery of DNA structure by Watson and Crick in 1953. Both scientists had laid the keystone to microarray technology by elucidating the DNA double helix structure. Sequently, further knowledge in DNA was obtained by Marmor and Dorey in 1961 as they found out that DNA could be separated by heat or treatment with alkali. Therefore, they were first to describe the DNA renaturation or molecular hybridization. Afterwards, Southern blotting were introduced by Edwin Southern in 1975 for improving DNA hybridization. This approach combines the separation of DNA based on fragment length by electrophoresis with subsequent hybridization of a probe for the specific detection of DNA fragments. Next, the concept of microarray began with the ability to analyze multiple hybridization targets in the late 1980s and early 1990s. This is when Hans Lehroche Group developed a pin-based robotic system and Steve Wader developed scanner for reading output. Eventually, the first study to use the term microarray was published in 1995 by Mark Shina. He and his co-workers developed a microarray technology at Stanford, where they demonstrate how this new technique could be used to track the expression of many genes in parallel. And finally, in 1997, the first whole genome of an eukaryotic species completely investigated by a microarray. So Saccharomyces cerevisiae is the first whole yeast genome that were investigated at that time. From then on, microarray has become a general term for many types of this technology, such as protein microarray, tissue microarray, and many more. And the most common type of microarray is DNA microarray. So let's take a look on what is DNA microarray. DNA microarray is an orderly arrangement of thousands of identified sequence genes printed on impermeable solid support, usually glass, silicon chips, or nylon membrane. And every spot on the chip represents a different coding sequence from different gene, and it is made up of DNA probe that can pair with the cDNA, which is the complementary DNA that was created during the experiment. And usually, a single DNA microarray chip, like the image on our right side, may contain 1,000 of spots, each representing a single gene and collectively the entire genome of an organism. And this enabled the researchers or scientists to perform a simultaneous analysis of literally thousands of genes in a massively parallel manner, or even entire genome of an organism, like bacteria, yeast, mouse, or even human in a single experiment. So this next image is to show how small the spot on a microarray chip. So each spot will contain a DNA probe.
So alright, I am Syalfira and I will continue with the principles of the DNA microarray and a little bit about the techniques used in DNA microarray technology. So first of all, the core principle in microarrays analysis is hybridization between two DNA strands. And DNA microarray technology is based on the fact that complementary sequences of DNA can be used to hybridize immobilized DNA molecules, which are referred to the DNA probe. And there are four principles in performing microarray experiment, which are: the first one is sample preparation and labeling, the second one is hybridization, the third one is washing, and the last one is image acquisition and data analysis. The techniques used in the preparation of the DNA microarray technology include 5 steps. The first step is we have to prepare microarrays by spotting gene-specific oligonucleotides that can be synthesized that are complementary to the RNA transcripts of every gene in organism's genome, and they can attach to solid supports such as nylon membranes, glass slides, silicon surfaces, or on arrays of microbits for use of hybridization probes. Next is the second step. RNAs to be analyzed are isolated from the cells or tissues of interest, for example, normal cells and cancer cells, and used to prepare cDNAs labeled with different fluorescent dyes. Next steps will be further discussed by my friend Ain Syazwani.
So the next step, to distinguish between the two pools of synthesized cDNAs, the molecules are fluorescently labeled using real-time PCR. Red for the tumor-derived pool and green for the cDNAs direct from the normal tissue. In the fourth step, the equal amount of cDNA samples are mixed together and hybridized to microarrays. These labeled cDNA are hybridized to the probes of microarrays to compare the level of expression of genes of interest or of all the genes in the genome. After hybridization is complete, cDNAs that are complementary to oligonucleotide on the microarray will bind or hybridize with the DNA and thereby stick to the location on the slide. The unbound cDNA are washed away. Lastly, in the last step, the arrays are then scanned with the lasers and fluorescent detector with micrometer resolution, and the results are analyzed and recorded using computer software designed to remove background noise and amplify positive signals. A scanner detects patterns of hybridization by sensing the fluorescent signals. In this example, a red spot indicates that the expression of the gene is higher in the tumor tissue compared to normal tissue. In contrast, a green spot indicates the expression of a gene is higher in normal tissue than in tumor tissue. A yellow spot indicates that the gene is expressed equally in both tissues. If the gene is not expressed in either tissue, the spot will not fluoresce because each area of the microarray contains a known DNA sequence corresponding to a known gene. The identities of the hybridizing cDNAs can be determined using this data. Investigators can establish which genes are expressed differently in the cancerous tissue and thus may be able to design better treatment strategies. This picture shows microarray hybridization data comparing the levels of expression of 588 genes in untreated human cancer cells and human cancer cells treated with a chemotherapeutic agent. Change in levels of gene expression induced by the chemotherapeutic agent can be detected by comparing the two arrays. So from this expression, gene expression signatures have been identified that are useful in clinical decision-making. Patients with a good prognosis can avoid unnecessary chemotherapy, whereas those with a poor prognosis can receive more aggressive treatment. To conclude, the ability to analyze the expression of entire genome has resulted in a vast amount of new information in biology and will eventually lead to an understanding of the normal process of human development and the causes of at least some human disease.
Next, we move to the uses and applications of DNA microarray. The first application is gene expression profiling. In mRNA or gene expression profiling experiment, the expression levels of thousands of genes are simultaneously monitored. This is because to study the effects of certain treatments, disease, and developmental stages on gene expression. It also can be used to identify genes whose expression is changed in response to pathogen or other organism by comparing gene expression in infected to the in uninfected cells or tissues. So the diagram shows of gene expression profiling, a clinical diagnosis of chronic lung allograft dysfunction disease. The second application is drug discovery. There are 4 steps in drug discovery. The first step is called target identification. This step is to discover the genetic basis of disease by mapping disease genes with whole genome SNP assays. Next, step 2 is target validation. This step is to pinpoint disease-causing mutations or to measure the genetic variability of a target gene in clinical populations. Then, step 3 is compound screening. This step is to characterize seed compounds for selectivity and specificity, and also to identify molecules that disrupt expression of intended disease gene. Last step is clinical trials, helping researchers predict which drugs will work for which patient with which disease. For an example, the red pill called Iressa is the therapeutic drugs for lung cancer disease. Thank you.
Another important application of microarray technology is as a comparative genomic tool. Gene mutation analysis is used to analyze genomic alterations such as sequence and single nucleotide polymorphism. In the context of microbiology, microarray gene mutation analysis is directed to characterization of genetic differences among microbial isolates, particularly closely related species. For example, detecting the presence or absence of DNA sequences or genes between pathogenic and non-pathogenic strains of the same species. Such informative approach would allow us to reveal genes exclusively present in the former that may be required for infectivity, virulence, or adaptation to a particular host niche. DNA microarray technology can also be used to compare between a fully sequenced genome and unsequenced but related genome of closely related bacteria. This kind of approach can provide us available information about the diversity and evolution of pathogens and symbionts. Comparisons of this kind use a microarray containing representation of all the open reading frames of the sequence, reference strains, and labeled DNA from the unsequenced experimental strain. It will then reveal genes common to both strains and genes that are present in the reference strains but absent in the experimental strain. However, this method may not detect genes present in the experimental strain but missing in the reference strain, although the use of multi-genome may detect genes present in the experimental strain. It may not also detect point mutation, including frame shift mutation, small deletions, and deletions in homologous repetitive elements.